Good afternoon from Los Angeles, California. My name is Babak Kateb, and I am the president of a Brain Mapping Foundation and also the chairman of the board for Society for Brain Mapping and Therapeutics. Today, I'm going to talk about the future of clinical neuroscience and also the integration of neurosurgery, AI, machine learning, neurophotonic brain mapping with cellular therapeutics. So before I go further on this, I would like to thank our dear colleague and friend, Mr. Bernie Siegel, for his amazing work in this area of research for stem cell and his fantastic work that he has done on this World Stem Cell Summit for many, many years. Okay, so let me start by also talking about series of organizations that are going to be represented here, Brain Mapping Foundation and the National Center for Nano-Bio-Electronics. I would like to make a disclosure here that I'm chairman of the board and CEO of Smart Microscopy, as a company that is focused on basically intraoperative brain mapping. In this case, you could see a real-time skull-based operation looking at brain cancer, and we are trying to use new generation of microscopes to intraoperatively detect brain tumors during the operation using multispectral imaging.
The other side, this is the left side, the right side. Okay.
So in this video, you see that it's very hard to delineate marginal brain tumors, and based on anatomical structure, we could see if this is a brain tumor or a normal cell or normal tissue. So you will see later on in my talk how we actually use AI machine learning to auto-delineate these tumors. The learning objective of my talks are fivefold. First, what is brain mapping, and what are Brain Mapping Foundation, Society for Brain Mapping Therapeutics, and Brain Technology Innovation Park, and also what's the definition of brain mapping, what are the examples of it, how new technologies can help applications and outcomes of stem cell, and what should be the future directions of stem cell research and development, as well as allocation of fund be. The Society for Brain Mapping and Therapeutic is a multidisciplinary organization, and it's focused on improving the diagnosis, treatment, and rehabilitation of patients afflicted with neurological disorders. As I mentioned, our approach is multidisciplinary in collaboration with government agencies, patient advocacy groups, educational institutions, and private sector industry, as well as philanthropic organization. Our next World Congress for Brain Mapping and Therapeutics will be at the LA Convention Center this coming July in person, and also virtual, that's optional. And we have about 900 presenters, about 12 keynotes, including former Secretary of the VA and Dr. Deepak Chopra, and some of the top scientists, including Eric Kandel, who is a Nobel laureate. So getting to definition of brain mapping, the study of the anatomy and function of brain and spinal cord through the use of imaging, that's including intraoperative imaging, microscopic imaging, endoscopies, and multimodality imaging, as well as immunochemistry, biochemistry, and molecular and optogenomics, stem cell and cellular biology, engineering, which is material, electrical, and biomedical, and neurophysiology and neuroanatomy. In a sense, brain mapping is like a Google map. You get a snapshot of Earth, that's your MRI. You zoom down all the way to cities, that's functional MRI. You can see the function of the cities. And then you zoom down on the streets, those are diffusion tensor imaging, tractography. And then you zoom again on the cars on the road, those are cells, people on the car are nanostructures or genomic materials. So brain mapping really focused anything from imaging all the way down to nanomedicine. And well, there are a couple of other definitions I have to go over here. What is nanoneuroscience, nanoneurosurgery, and nano-bio-electronics? Application of nanotechnology in neuroscience is called nanoneuroscience. The therapeutics application of technology, nanotechnology, which could enhance neurosurgical treatment, is called nanoneurosurgery. And integration of nanotechnology devices, cellular therapeutics, and imaging is called nano-bio-electronics. Well, Society for Brain Mapping and Therapeutics has been around for about 18 years, and we have health conventions globally, and it's kind of a growing organization. It's a multi-specialty association. Here you can see I get a letter of appreciation from the mayor of Los Angeles, and as I said, we are holding our 17th and 18th annual convention in July of this year at LA Convention Center. Through this organization, we have basically published series of special issues with neuroimage, and we have published series of, two textbooks, inaugural textbooks, one on neurophotonics and brain mapping, one in nanoneurosurgery, which I presented to President Obama, and he thanked me for my effort and in regard to this book. We have partners with SPIE, a special issue with the neurophotonics, and also published a special issue with PLOS ONE. We have been deeply involved in global policy and neuroscience from President Obama's Brain Initiative, which I was advising the White House at that time, and attended the Brain Initiative announcement at the White House with the Honorable President Barack Obama. And also we educate the head of NASA here. You can see is standing with me, and Supreme Court Justice Elena Kagan. I've spoken at the United Nations, at the Australian Parliament, and Canadian Parliament. We started a series of brain initiative globally, also Malaysian Parliament, and also we started the IEEE Brain, and in the process, of course, we awarded Stephen Hawkins and the co-chair of the Congressional Neuroscience Caucus, Mr. Blumenauer. As part of our Global Brain Initiative, we started what's called Neuroscience 20, which is Brain 20, Spine 20, and also Mental Health 20. The focus of this initiative is on the cost of neurological disorders to the world economy. You all may know that neuropsychiatric disorders in the US alone approach about $1.5 trillion in expenses, and which 200 billion of that is related to Alzheimer. And the cost of traumatic brain injury to the US economy is approaching about $100 billion. So we're looking at the global collaboration through the G20, and we've been talking to the European Union president and other delegations from China, Turkey, Germany, and so on in the last several years to be able to collaborate and, in a sense, um, you know, reduce the cost of healthcare and increase efficiency. Some of the goals of the G20 Summit are G20 Summit's Neuroscience 20 are focused on formation of national data repository and analysis for neuroscience, creation of national network of brain procurement, allocation, and banking, establishment of a global alliance for Nano-Bio-Electronics, which we can actually integrate stem cell with other platforms for brain mapping and therapeutics, and facilitate rapid introduction of diagnosis and therapeutics to address neurological disorders, which is going to be done through our next initiative, which is a Brain Technology Innovation Park, which is located in Los Angeles. And the aim of the park, again, is to tackle the cost of neurological disorders by introducing game-changing therapeutics and diagnostics. We are achieving this through global partnership, and the organizations that are on the lead are Society for Brain Mapping and Therapeutics, with about 5,000 members globally, and also Brain Mapping Foundation, which is the charity arm of the organization, and under the umbrella of Neuroscience 20. So the Society for Brain Mapping has an interesting story that we started this as a small summit about, I would say, more than 15 years ago at JPL in partnership with USC and Caltech, which has grown to, basically a very successful kind of global collaboration. We have created a kind of an ecosystem for Brain Technology Innovation Park in partnership with service providers from law firms to accounting and so on, to investors, and also national labs and large companies, as well as universities and Department of Defense and government agencies. So this was the step one of the kind of phase one of the Brain Technology Innovation Park. And now we are actually moving to the phase two to create the physical space for it. So the question is, okay, we do all this initiative, but then what is coming out of it? And the answer lies in the next several slides that I'm going to show you, which will include also some stem cell research and therapies. Well, I would like to draw your attention to physical properties of waves in this slide. And in this slide, you see that we are looking at the ultraviolet imaging in 10 to 400 nanometers, and infrared about 700 nanometer to a meter, and microwave, which is 1 millimeter to meter. So 1 meter. So let me start with microwave ablation. So we actually worked on this device for about 15 years to design a device to be able to actually microwave cancer. But not only doing that, you could actually heat up the blood-brain barrier to the level that you can actually deliver nano drugs to it. So in this case, you can see in this picture on the left lower side, this is the MRI of a pig brain that we actually ablated part of the brain. And you can see a very round ablation, the histology and the gross anatomy of this animal model shows the proper lesioning by this microwave ablation technology. So here I'm going to show you a minute of video, and this device was FDA approved. So you could take a look at how this technology could actually help in the future of brain cancer therapy.
All right, this is a device, microwave ablation device that we have microwave with a chokehold, and can create actually a lesion for the size that you want. The device is actually controlled by the computer, any laptop, and the plan is that at the time of a biopsy, we basically put the biopsy needle out and the microwave ablation in, and basically ablate the brain tumor.
So it's very simple. At the time of biopsy, as I said, you can put this the probe in and dial the size of the lesion and do the microwave ablation of brain cancer.
All right.
So there are other technologies that I would like to bring to your attention, and that is nanotechnology. So in this case, we use NASA's carbon nanotube for cytokine and drug delivery to brain cancer using macrophages. So we took the carbon nanotubes and broke the kind of introduced the sulfur in between carbon-carbon bonds and basically cultured them with a poronic dye, which turned them to a kind of a red autofluorescent particles, which could be picked up by macrophages, and macrophages break down the carbon nanotubes, and cytokines will be released and activate them for cancer therapy. So these are live human macrophages that have picked up the actual carbon nanotubes, which were developed by NASA Jet Propulsion Laboratory. So in the slide on the left side, you see that these cells are actually tolerating these carbon nanotubes, and the cytokine level kind of goes up, but normalized. And the ratio of DNA and siRNA you could actually load up in these carbon nanotubes are 1-to-1, which allows you to actually calibrate your therapy. It's the same platform now being used kind of in animal model. The red dots are basically multi-wall carbon nanotubes, and the green dots are CD68 cells, macrophages, macroglias, and the blue cells are basically tumor cells. You can see that these cells are traveling and kind of attacking brain tumors. Well, one of the issues with stem cell therapy is that how do you actually visualize the cells that you are already transplanted into someone's body? You basically you're injecting millions of cells into someone's body, and you would like to see where they are going. So one of the solutions to this is through basically AC magnetic field, stimulated nano electroporation of magnetoelectric nanodrug couriers inside the CNS in this case. So you could actually do magnetic electroporation to insert the magnetic nanoparticles in the cells, and you can actually basically image these cells and track them. In this case, basically we did this in animal model, in mice model, but also did it in in monkey model. The results are promising, and we could track these cells and the toxicity of these magnetic nanoparticles seem to be kind of very low, and it could be very good tools for cellular tracking. Other nanoparticles include basically nanoprodrugs. In this case, you could see ibuprofen is used, and there are spacers that we have created, and in between these spacers, alpha tocopherol, and also another spacer, in this case, one octa decan ethiol, are used. So these create a micelle type nanoparticle that you could actually tag them with cis 5.5, and basically that's a melamide fluorescing tracer, which you could actually or tag them with gallium with a different kind of uh, I would say contrast agents or intensity. So you could actually track cells with these nano prodrugs, but also deliver uh, um, a therapy to the um, area that you are targeting. Well, let's just focus on a variety of different applications that this nanoparticles could have for Alzheimer drug delivery, for stroke and chemotherapy. But in this case, you can see the nanoparticles have gone to the area of traumatic brain injury. And as I mentioned in the past slide, we have used ibuprofen, which is anti-inflammatory, and alpha tocopherol, which is antioxidant, and that could be used for reducing inflammation, increasing oxygenation in the area of neurotrauma. So in the future, you're going to see paramedics giving a bolus of nanodrugs to neurotrauma patients, stabilize them before they get to the ER and the hospital. Other applications of nanotechnologies basically are in electronic nose. So in this case, we have used electronic nose to sniff cancer cells. So these are nano films that trap compounds and give you a signal. We kind of published this in Neuroimage back many years ago, but now same concept could be applied for actually detecting COVID-19, and we received an award from NASA. Well, some of the applications of AI machine learning and predictive modeling could be actually used for visualization and modeling the spatial distribution of a neural stem cell within intracranial glioma. As you know, neural stem cells have tumor tropic properties. And in this case, we have used mathematical modeling in order to predict therapeutic efficacy of representation of the neural stem cells based glioma therapy, which was published. And the point here is that it is very important to use mathematical modeling and predict the pattern of distributions of cells, stem cells, and tracking them with the nanoparticles in terms of future therapies for brain cancer and other neurological disorders. Again, this is a beautiful 3D visualizations and quantification modeling of neural stem cell distributions for brain mapping and therapeutics. You can see the red dots are neural stem cells, and the green area is brain tumor. And so you could actually use nanoparticles and track cells, but also use mathematical modeling, looking at the distribution of stem cells that you're trying to target to certain areas. And of course, other technologies that come to play for future of brain tumor therapy that could be also integrated with stem cell and vice versa is multispectral imaging. So imagine multispectral imaging as kind of a a bunch of tennis balls that you're throwing at the wall or a curtain in your house, the tennis balls bounce back from the wall pretty directly at you, but if you're throwing a tennis ball to a curtain, it actually drops down. So these photonic particles actually bounce back from normal cells and cancer cells with different speed and decay over time. So because of that, you could use multispectral imaging and for brain tumor detection. So in this case, you see kind of real-time intraoperative resection of a malignant melanoma metastasizing to the brain. Melanomas are skin cancer, and they can metastasize to brain and form a kind of a tennis ball shaped growth in the brain, which is highly vascularized. In this case, it's very vascularized. You see surgeons are trying to take this out. But it's very important to be able to actually detect the margin of this and take out 100% of the tumor. But you will see here the tumor is out, and they're going to suction the tumor cavity. In this case, with the visual, kind of visible light and visual, just the naked eye, you would think there's nothing left. Now, let's take a look at the same procedure in infrared. That's the tumor. These are the retractors, and this is the vessel nearby. The surgeons are taking it out, and you can see the tumor is taken out, and very quickly you will see the tumor in the petri dish, despite of the fact that there are no vessels attached to it. The thermal signature is higher because of the metabolic activities, we think. But then you see a little bit of higher thermal signature here, which later on we confirmed that there was a little bit of potential residual tumor. This was published in Journal of Neuroimage. So the application of multispectral imaging, including thermal imaging, into brain mapping is really, really helpful for future total resection of brain tumors and brain tumor therapy. Another application that comes to mind is, in this case, virtual reality. This is virtual reality brain mapping of the skull-based tumor. You'll see that the tumor here is in the color of blue, and you could actually rotate this kind of imaging and then walk through it, so we can visualize where the tumor is in your mind, so you can have a better tumor resection, and you can kind of do your surgical planning according to this type of technology. In this case, it's this surgical theater technology. So another multispectral imaging system is basically ultraviolet imaging that we use with NASA, and in this case we have integrated these technologies all together and use AI machine learning for kind of real-time intraoperative brain mapping, and used multispectral imaging, as I mentioned, because some of these fluorophores upregulates in brain cancer, and you can actually use multispectral imaging to detect where the cancer cells are. In this case, all the yellow spots are cancer cells, and all the blue dots are normal cells. Same concept could be applied for stem cells. You can actually use multispectral imaging to detect, mature stem cells and so on. Of course, AI machine learning these days is very attractive. We are using AI machine learning for kind of intraoperative and overall brain mapping and therapeutics. And this could be applied for stroke, dementia, tumor, and multiple sclerosis and other kind of disorders. And the data could be, kind of visual data and big data and so on. So this data could be kind of pancaked and create a 3D model. In this case, that could be really help us for auto delineation, as I said, and could give you a beautiful high resolution, kind of tractography, looking at the highways of the brain, so you can actually avoid cutting through these highways. But at the same time, I would like to actually draw your attention that when you're implanting cells into the brain, stem cells, it is very important that we actually be able to connect these connections that are missing, let's say in the Alzheimer patients. And that is something that I would like to draw your attention to, because one of the biggest challenges in stem cell therapy is the integration of stem cell with the circuits of the brain, at least in the neural stem cell, and that's one of the challenges. So again, this is another multispectral imaging that you see for glioblastoma multiforme. It's very hard to delineate the tumor, but you can see with multispectral imaging, you can see these tumors in beautifully light up. So you can achieve total resection. And with machine learning nowadays, we could actually do a predictive modeling of these tumors and basically predict the pattern of behavior of these tumors for intraoperative tumor mapping and vascularization. So you could actually predict and prevent the growth or do the adjunctive therapy in order to have a better outcome for the patients. Well, these technologies and platforms that I presented to you are in partnership with our colleagues at the Oak Ridge National Lab or Lawrence Livermore and AI Gnostics and NASA and the SBMT. And in conclusion, I just want to say that the integration of nanotechnology, AI, VR, AR, supercomputing, multimodality, brain mapping, imaging, cellular therapeutics, neurophotonics, and or devices will be the future of brain mapping and therapeutics. So I think this is a great World Stem Cell Summit, but I think I want to draw your attention to the fact that the future of cellular therapeutics is in the integration of cellular therapeutics with other modalities or vice versa. How do we acquire data is very important. So I think AI machine learning should play a significant role in the future application of stem cell. So how do we share it and store this data, and how do we use this data going to impact the AI reliability? But integration of such data will, with cellular therapeutics will impact the outcome of the stem cell therapies of the future. I also encourage that all of you to be more involved in policies. As you saw, as an organization, we are very involved in policies and formation of new initiatives. I know our Bernie has been kind of a force of nature in stem cell policy. And I truly admire him and the World Stem Cell Summit for their fantastic work that they are doing in this area. And we are looking forward to collaborate with them. I also have a recommendation for CERM and Proposition 14. to focus their funding toward integration of other platform with cellular therapeutics. In case of nano material for cellular stem cell cellular tracking, or AI machine learning application for predictive modeling for distributions of the stem cell throughout the tumor cavities and so on. So I think this area could provide a significant number of solutions for neurological disorders and other disorders. And at the end, I just want to invite you to the 18th Annual World Congress of SBMT. And again, I would like to thank my wonderful friend Bernie for providing me with this great honor to be part of this magnificent organization, World Stem Cell Summit, and and the foundation. So looking forward to seeing you in Los Angeles, and thank you so much for your amazing work that you're doing. Thank you.